All right, welcome back. So in this module, we are going to look a little bit more closely at chromosomes. So you know that the genetic information is contained in these chromosomes. So let's talk about them. A chromosome is composed of coiled DNA. In eukaryotic organisms, the DNA is wrapped up by these proteins that are called histone proteins or histones. So I have two examples or models of chromosomes, if you will. Over here on the right, you will see a typical eukaryotic chromosome, and you have seen a picture like this before. Actually, this is two chromosomes, two identical copies of one chromosome that are connected at that spot called the centromere. So this is what chromosomes look like right after DNA replication, where they consist of two identical copies of the same DNA molecule. So over on the left, I have a bacterium right here is a bacterial cell. And inside of that bacterial cell, if you will remember, bacterial chromosomes are a little bit different in structure. Here you see that a bacterial chromosome isn't in those long linear chains like the eukaryotic chromosomes are. They are instead coiled up into a long loop that is connected at both ends. They have a single circular chromosome. Many bacteria also, however, carry these little extra bits of genetic information. Those are called plasmids. So I want to talk about the eukaryotic chromosome a little bit and how it's organized. So if you start down here at the bottom of this figure, you will see an entire mitotic chromosome. And again, that's just a fancy way of saying that this chromosome is actually composed of not one, but two copies, identical copies, of the same chromosome. So what I want you to imagine is that you have a magical microscope that can zoom in on this one little tiny section of the chromosome. What you would see if you zoomed in on that little tiny section is a bunch of what look like wet ramen noodles. Now, if you were to continue to zoom in on these little noodles, what you would see is what looks like a jarbled up bunch of beads. Each of those beads, if you were to look a little bit more closely, you would see those beads right here. Those beads are actually not DNA. Those are called histone proteins. And again, those are the proteins that coil up the DNA into the nice, neat package that we call a chromosome. So again, if we zoom in even further, you see that that histone protein has wound up this blue stuff. That blue stuff, if again you could zoom in on a small segment of it, you would recognize that immediately as the double helix of DNA. So ladies and gentlemen, from the super duper small, zooming out to a much larger view, you see how DNA is organized into these chromosomes in a eukaryotic cell. Now in the prokaryotic cell, those chromosomes are kind of just floating around in the center of the cell. And again, we call that the nucleoid as opposed to a true nucleus, which a eukaryotic cell uses to hold all of its chromosomes in. So there you have them, the chromosomes.